So here we have a brief outline to summarize what we have discussed thus far. We discussed the intermediary fuel molecule ATP, and that ATP is a molecule that contains energy in its phosphate bonds. And the energy was transferred from the oxidation or quote unquote burning of a fuel molecule. ATP, once produced, is then hydrolyzed through ATP hydrolysis, which is essentially a reaction that splits open the last phosphate bond, releasing the energy. In the context of muscle contractions, that energy is transferred to the myosin head so it can do work on the thin filament, sliding it across to produce a muscle contraction. We began talking about the bioenergetic systems or ATP producing systems by discussing the anaerobic systems first, which again is simply ATP producing systems that kick in at intensities above the anaerobic threshold, where the aerobic system cannot fully meet the ATP demand because the oxygen availability does not meet the increasing oxygen demand. Now, there are two anaerobic systems, again, that do not require oxygen and are generally fast ATP producers. The first anaerobic system we introduced was the phosphagen system, aka the ATP PCR system, aka the lactic system. This is the fastest ATP producing system. Why? Because it only requires one reaction to produce one ATP from one phosphocreatine, and this is catalyzed by which enzyme? Creatine kinase. Also, although the phosphagen system is very fast at producing ATP, it is highly limited in capacity. It is limited in that you only get one ATP per fuel molecule, which again is phosphocreatine. And also there is limited phosphocreatine stores in the muscle cell at any given time. In fact, as we discussed, phosphocreatine can be fully depleted in just 15 seconds of all-out muscular work. Now, we said that the phosphagen system is most active during post-anaerobic threshold intensities, but mostly during maximum intensity exercise, so all-out efforts. We also said that the phosphagen system is temporarily highly active during the onset of exercise until the cardiovascular system has enough time to deliver sufficient oxygen to the muscle. At that point, the aerobic system can fully meet the ATP demand, and the phosphagen system's activity decreases at that point. I also went into discussion about the role of nutrition in the phosphagen system. Now, there is evidence to suggest that supplementing the diet with creatine can enhance phosphocreatine stores in muscle, which can in turn translate to increased performance by increasing exercise capacity, especially during high to max intensity exercise bouts, or in other words, post-anaerobic threshold exercise bouts. Now, moving on, we said earlier that there are two anaerobic systems that again produce ATP fast and produce it without oxygen. Again, these anaerobic systems kick in when the aerobic system cannot fully meet the ATP demand of the working muscle. I know I'm redundantly saying this, but there is a reason I am saying this over and over again, because it is a point of emphasis as you prepare for the exam. So like all anaerobic systems, this system is also a backup system to the aerobic system. Again, there's never a situation where the aerobic system completely shuts off and the anaerobic system takes over. Aerobic systems are always active, and anaerobic systems only cover the portion of the ATP needs that the aerobic systems cannot cover. This occurs during post-anaerobic threshold exercise. So the second anaerobic system is called the lactic system, or fast glycolysis. These two terms are used interchangeably. Many also just refer to this as simply glycolysis. So what is fast glycolysis? Well, fast glycolysis is described as rapid and partial carbohydrate oxidation. Again, rapid and partial carbohydrate oxidation. Please remember this. Also remember, oxidation in very simple terms is the process by which a fuel molecule is broken down to release its energy, which is used to form ATP, which contains that energy briefly until it is hydrolyzed, like on the myosin head during muscle contractions. So the other key words here are rapid and partial. Rapid, meaning this is fast carbohydrate breakdown, meaning ATP is produced rapidly, similar to the phosphocreatine system, and it is partial carbohydrate breakdown, meaning the carbohydrate fuel molecule is not fully broken down after glycolysis, and that at the end of glycolysis, there is still energy left in the fuel molecule. Now remember before, as we discussed the phosphagen system, the rate at which ATP is produced from a single fuel molecule depends on the number of reactions involved. The more reactions, the more time it takes to yield ATP. So fast glycolysis requires 10 reactions to break down the carbohydrate fuel molecule to produce ATP. This is still very fast, but not as fast as the phosphagen system. Why? Well, again, because fast glycolysis has 9 more reactions than the phosphagen system. But here's the deal. Although it is slightly slower than the phosphagen system, you get more ATP per fuel molecule than the phosphagen system. The phosphagen system yields only 1 ATP per fuel molecule, which is phosphocreatine. The lactic system, or fast glycolysis, yields 2 to 3 ATP per fuel molecule, which is carbohydrate. So what type of carbohydrates are we talking about? Remember, these are your endogenous carbohydrates. We have 2 types of endogenous carbohydrates. One is the simplest carbohydrate form called glucose, and glucose exists circulating in the blood. The major sources of glucose is from your diet. So when you eat carbohydrates and your digestive system breaks it down into glucose, your blood glucose levels will rise. Glucose can also be produced in the liver through a process called gluconeogenesis, formation of new glucose. Once the liver produces it, it is released into circulation. So glucose levels in the blood is maintained at around 70 to about 100 milligrams per deciliter of blood. This is called blood glucose homeostasis, and throughout the day, different cells in the body will take it up. When there's a large uptake of glucose by cells, the blood glucose levels will drop, and the body will ramp up production to keep it maintained at homeostasis levels. So here we have an example of glucose in circulation, and the muscle cell is taking it up in a process called glucose uptake. So now the glucose is inside the muscle cell, and this glucose can now be used as a fuel source. The glucose can also be stored up if it is not immediately used as a fuel. It can be stored up as glycogen, which I said was like a bunch of glucose molecules bonded together. So the other form of endogenous carbohydrate is glycogen, which again is simply a stored form of carbohydrate. It is largely found in skeletal muscle as well as the liver. But the difference is that in the liver, glycogen is used to form glucose, meaning in the liver, glycogen can be broken down to make glucose. In the muscle, however, glycogen is not used to make glucose because it can't. The muscle does not have the required enzymes to form glucose with. The glycogen in muscle is broken down to provide a fuel molecule for the muscle. So here is an important point that I will reiterate later. The starting point of glycolysis is a molecule called glucose 6-phosphate. That's glucose 6-phosphate. And glucose 6-phosphate can be derived from either glucose or glycogen. Glucose only has to go through one reaction to be converted to glucose 6-phosphate. Glycogen has to go through multiple reactions in a pathway called glycogenolysis, lysis, or breakdown of glycogen. Through glycogenolysis in muscle, you form glucose 6-phosphate. So technically speaking, glucose 6-phosphate is the fuel used by the lactic system to produce ATP. And again, glucose 6-phosphate can be derived from glucose or glycogen. This is why glucose and glycogen are your muscle's carbohydrate fuel sources, because it is a source for glucose 6-phosphate, which again is the starting point for glycolysis. More on this in a bit. So first, before we start discussing fast glycolysis or the lactic system, let's talk about glucose uptake and how glucose gets into the muscle cell. Whether it is used to form glucose 6-phosphate to enter fast glycolysis or to be stored up as glycogen, glucose first needs to somehow get inside the muscle cell. So this process is mediated through what we call a signaling pathway. And just like the term implies, it is simply a pathway of signals that eventually result in some cellular process like glucose uptake. So let's first discuss the signaling pathway shown on the right. Now, as I said before, normal blood glucose levels range between 70 to about 100 milligrams per deciliter of blood. So whatever your normal blood glucose is, say it is 80 milligrams per deciliter blood, that is your body's homeostasis level. That is where your body wants to keep your blood glucose levels. Now, how do you think you can perturb this? What can you do right now to temporarily raise your blood glucose levels up and get it out of this homeostasis/normal level? What you can do is eat food, more specifically, eat carbohydrates. Like I said earlier, when you eat carbohydrates, no matter whether it is a complex carb inside some pastry you like or something simple like table sugar, your digestive system will break it down into its simplest form. And that simplest form is mainly glucose. There's also fructose, but let's just keep it simple and focus on glucose. So after the digestion of the carbohydrates you consumed, you have glucose, which will then be absorbed into circulation. This is the point where blood glucose rises. This is referred to as an acute hyperglycemic effect, or simply acute hyperglycemia. Acute just referring to the fact that hyperglycemia is temporary. This means you are temporarily above your homeostatic normal range. Now, what we know about the human body is that when homeostasis is disrupted, like in this case, it will respond to reestablish homeostasis. So your body will respond to this blood glucose levels rising after you ate some carbs. It will respond in a way that will lower it back down to your homeostatic normal range of 80 milligrams per deciliter of blood. So the question now is, how does your body do this? How does it bring your blood glucose back down to normal levels? The primary mechanism for lowering blood glucose is through a powerful hormone called insulin. Insulin is produced and secreted by the pancreas. The pancreatic cells, also known as beta cells, that produce and secrete insulin, quote unquote, senses when blood glucose levels rise above normal levels. When it senses a rise in blood glucose levels above homeostatic levels, it will secrete insulin into blood circulation. The insulin will then circulate to different cells in the body where it will interact with the cells and function as a signal. So here we have a muscle cell, for example, and on the membrane of the muscle cell, or the sarcolemma, we have what is called a insulin receptor. As the term implies, it is a receptor that binds to insulin. So as insulin circulates, it can bind to its receptor on various cells in the body, like the muscle cell in this example. Once it binds to the insulin receptor, it will initiate a signaling pathway as shown here. Now here I portray the signaling pathway with a single arrow, but it is much more complex than this. There is a series of processes within this signaling pathway. But for the sake of simplicity, just know that when the insulin binds to its receptor, it will initiate a signal down a pathway. And what does this signal result in? It results in the activation of a protein called the GLUT4 transporters, as depicted here by this blue figure. When the GLUT4 transporters receive this signal, it will physically move towards the membrane of the cell. So it will physically move to the sarcolemma. This is called translocation. So in response to insulin binding to its receptor on the muscle cell and the initiation of this signaling pathway, GLUT4 transporters will translocate to the cell's membrane. Once it reaches the membrane, it will dock itself to the membrane and become sort of an access way for glucose in the blood to enter the cell. So when GLUT4s are present on the cell's membrane, as shown here, glucose that is circulating in blood will then come inside the cell. This is again called glucose uptake. So when this occurs, obviously the blood glucose levels will decrease since the glucose is now out of the blood and now inside the cell. In this example, the muscle cell. Eventually, the blood glucose that increased following the carbohydrate meal is now lowered back down to normal levels. So for example, if blood glucose homeostasis was 80 milligrams per deciliter, and after you ate, it rose to 112 milligrams per deciliter, your pancreas will quote unquote sense this rise in blood glucose and will release insulin to lower it back down to 80 through this mechanism we just described here on the right of this figure. So now that glucose has come into the cell, it can be used by the cell as fuel. It can be converted to glucose 6-phosphate to undergo oxidation, or it can be stored as glycogen for later use. So is there another route for glucose uptake? There is, but only for muscle cells. And this is one of the reasons why muscle is so unique compared to other cells in the body in the context of glucose uptake. One of the unique aspects of muscle cells is that it has the ability to contract and produce muscle tension and force. No other cells in the body have that ability. And what else is unique about muscle cells is that glucose uptake can be stimulated through muscle contractions. This is referred to as mechanical stimuli. Muscle contractions, as shown on the left, can activate the GLUT4 transporters, which will then translocate to the membrane of the muscle cell, the sarcolemma, and bring in glucose from the blood and into the cell. Now, here is a critical point for you to take careful note of. The uptake of glucose in muscle cells via muscle contraction or mechanical stimuli is independent of insulin. You don't see insulin anywhere in this left side of this figure, meaning insulin does not need to be present or do anything in order for muscle cells to uptake glucose when it contracts. So what are the practical implications of this information? This means that when you exercise, there is increased glucose uptake in muscle cells, the contracting muscle cells at least. So if I ask the question, why? Well, it is because muscle contractions through mechanical stimuli stimulates glucose uptake, because these GLUT4 transporters are activated in response to muscle contractions, again, also known as mechanical stimuli. And again, this occurs even without insulin. This is why muscle is so unique in the context of glucose uptake. So if I ask the question, in what ways does exercise affect blood glucose? The answer would be that it lowers blood glucose. In other words, it has a hypoglycemic effect, HYPO glycemic effect. It acutely lowers blood glucose. Now, if I asked a true and false question to the effect of the decrease in blood glucose during exercise is due to increased insulin secretion, this is a false statement. It is due to the mechanical stimuli of GLUT4 transporters through muscle contractions. So let's dissect this even further. Since exercise through muscle contractions stimulates glucose uptake into the muscle cells, thereby lowering blood glucose, would you expect an increase in insulin during exercise? No, you would not. Why? Because insulin is only secreted when blood glucose rises, like after you eat some carbs. Now, this blood glucose lowering effect of exercise can be easily tested. You can get a glucometer, and through a finger prick sample of blood, you can test your blood glucose levels before exercise. Then you can do some high-intensity interval training, for example, and check your blood glucose levels right after your bout of exercise, and you will see that your blood glucose levels dropped a little bit. So again, why did the blood glucose drop during this experiment? It was because of the muscle contractions during the HIIT training bout, stimulating the GLUT4 transporters in the contracting muscles, bringing the glucose out of blood circulation and into those muscle cells. Now, if you think of the biological importance of this, it is pretty cool how the muscle is designed. When you exercise and contract your muscle cells need fuel for energy. So this process, as we just explained, is sort of an automated system for fuel uptake. The muscle cell, when it contracts, provides itself with fuel automatically, bringing in glucose in from the blood, simply just by contracting. Okay, so now what are the clinical implications here? Let's talk about type 2 diabetes. Now, type 2 diabetics are characterized by having high resting blood glucose levels, usually above 180 milligrams per deciliter of blood, which over time can cause organ or tissue damage in the body. So why do type 2 diabetics have high blood glucose levels? Well, it's because they have difficulty lowering it. They have difficulty lowering blood glucose because cells have difficulty taking it in from the blood. So, in other words, glucose uptake in cells is impaired. Glucose uptake in cells is impaired because of something called insulin resistance. Severe insulin resistance is the cause of type 2 diabetes. It's not because you ate too much candy or too much sugar, it's none of that. It's because of severe insulin resistance. So, what is insulin resistance? It is, in essence, a defect in this process right here on the right. There is a disruption in the communication between insulin and the insulin receptor. Thus, in those who have severe insulin resistance, like type 2 diabetics, this signal right here is not transmitted, at least very effectively. It doesn't mean that every single cell in their body is insulin resistant or every cell is completely insulin resistant. It's just that they have an increasing number of cells that are insulin resistant. And thus, overall in their body, this signal is not very well transmitted. So in type 2 diabetics, they consume carbs and blood glucose rises like usual. Also, the pancreas secretes insulin in response to this as usual. But the issue is that the insulin either can't bind to its receptor on various cells or the insulin can't communicate with the insulin receptor. Either way, the signal does not reach the GLUT4 transporters in the cells, and the cell has no mechanism for uptake of glucose. And again, this is relevant to most cells in the body, not just muscle cells. And this is why type 2 diabetics experience fatigue quite easily. Cells in the body like muscle cells and brain cells utilize glucose as a fuel molecule, and they are not able to uptake glucose very well due to insulin resistance. Now the next question is, why is exercise important for type 2 diabetics? We all know that when type 2 diabetics are diagnosed and go see their physician, what is the first thing the physician says? You need to exercise more. But here's the thing. Why the patient needs to exercise more is what is communicated less to the patient. Why do you think that is? Because believe it or not, many physicians don't know why. They don't know why exercise is important for type 2 diabetics. I remember taking my mother, who is a type 2 diabetic, to see her new physician. And the physician kept telling my mom to exercise more. And my mom was asking the physician on what type of exercise. And at this point, I can tell the physician was just making things up on the fly. So I interjected and asked the physician, why do you recommend exercise for my mom? How does exercise affect her condition as a type 2 diabetic? How does it benefit her? And the answer the physician gave me was that exercise will help her lose weight and that will help her with diabetes control. That was the wrong answer, and at this point I realized that my mom needs to find a new physician. That is not why exercise is important for type 2 diabetics. Let's explain using the information I provided thus far. So in type 2 diabetics, which side of this figure on this slide did I say was defected? The right side. The insulin resistance is the issue, meaning this right here is broken. But what did I say about the left side of this figure? I said that glucose uptake via muscle contractions is independent of insulin, meaning insulin is unnecessary for glucose uptake via muscle contractions and mechanical stimuli. So in type 2 diabetics, this mechanism right here is working just fine in their muscle cells. So even though the muscle cells are insulin resistant, there is another mechanism for glucose uptake that is still working for them. This means in type 2 diabetics, they can lower their blood glucose levels and therefore better control their daily glucose levels by simply contracting their muscles, causing glucose to be taken up by muscle cells and taken out of the blood. So the explanation that the physician should have given was this: exercising daily can help with daily blood glucose control for type 2 diabetics by lowering blood glucose. It can do this through muscle contractions. You see, when muscles contracts, this causes muscles to take the glucose from the blood and use it as fuel. The medication you are taking right now to manage your blood glucose levels is trying to do the same thing. It is trying to make the cells in the body bring that glucose out of the blood and utilize it. So literally, exercise is medicine. Exercise is doing the same thing that your medication is trying to do, but it does it without the use of chemicals. So this is why you should exercise on a daily basis so that your muscles can utilize that glucose that is flowing around in your blood and lowering that blood glucose through natural means. So I'm sure many of you at least know one person who is a type 2 diabetic. And now that you have learned this information, you can now explain much more comprehensively and intelligently as to why exercise daily is important for them. Okay, so obviously there are some important clinical implications here. But what else is there? What if you were a diabetic with a severe case and your pancreas was so damaged that it has trouble secreting insulin? At this point, your physician would prescribe insulin injections to help control your blood glucose. Now, what do you think would happen if you injected with insulin and right away you went and did some heavy exercise? Let's think critically. So after you inject it with insulin, there is going to be rapid glucose uptake by various cells in the body. Keep in mind that type 2 diabetics, they are not completely insulin resistant. Insulin still works, but just not efficiently. So when insulin is injected, the activity of this signaling pathway, as you see here, is high, and blood glucose levels will drop. And then you are exercising. Now, along with this pathway on the left is now very active, and thus you have two pathways of glucose uptake that are very active. What happens then? Well, at this point, you are at risk of severe hypoglycemia, which is blood glucose levels dropping too low, usually below 70, meaning your blood glucose levels can drop to a dangerous level because you have now two mechanisms stimulating glucose uptake. This is obviously very risky and could lead to an accident, especially since you are exercising. This is also relevant to most diabetic drugs, not just insulin. Many drugs stimulate glucose uptake by making these receptors much less resistant. So it is not recommended to exercise immediately after insulin injection or blood glucose lowering medication. It can lead to severe hypoglycemia. Once it gets to 50 and below, you are in trouble. Now, outside of my position here as a professor, I do a lot of consulting, especially for legal teams. There are a lot of lawsuits out there where a personal trainer or a gym is getting sued because a client who has type 2 diabetes and takes insulin injections or blood glucose lowering medication passed out, or in worst cases went into a coma during their session due to severe hypoglycemia. And they passed out or went into a coma due to severe hypoglycemia because they exercised immediately after their medication, whether it was insulin or the blood glucose lowering medication. And oftentimes when I review the case files as an outside expert consultant, it is the case that the personal trainer did not understand the information that I just went over. They did not know that exercise plus insulin or glucose lowering medication was a very bad combination because they did not understand the science as I just discussed. So the information that I discussed in terms of the mechanisms underlying glucose uptake and all the practical and clinical implications is an important piece of knowledge to have, especially as you go into medical and allied health fields, and especially if you go into the fitness fields working with clients who may be a type 2 diabetic taking blood glucose lowering medications. Okay, so the main takeaway here is that when glucose levels rise, say after you consume carbohydrates, insulin is secreted, and through this pathway right here will lower blood glucose back to normal levels by stimulating glucose uptake in cells. What is unique about muscle cells is that it has an alternate pathway for glucose uptake, and that is through mechanical stimuli, which is afforded through muscle contractions. So yes, exercise will have a blood glucose lowering effect. Okay, so now that we understand how glucose gets into the muscle cell, let's see how it is used as a fuel molecule to form ATP with through the lactic system, aka fast glycolysis. All right, so first you see glucose here in the blood, and this dotted line here represents the membrane of the muscle cell, and then beneath it is inside the muscle cell in the sarcoplasm, so the intracellular space. Now, as we discussed in the previous slide in detail, the glucose will enter the muscle cell and immediately undergoes a reaction that converts the glucose to glucose 6-phosphate, which is the starting point of glycolysis. And as you can see here, we have the ATP converting to ADP. What this means is that this reaction cost energy, and that energy came from ATP. So essentially, this first reaction expends 1 ATP. Now keep in mind, glucose 6-phosphate can be used to form glycogen to be stored for later. So it can go this way as well, from here to here. And this process is referred to as glycogenesis, formation of glycogen. Potential bonus question. Now we can also get glucose 6-phosphate from the breakdown of glycogen that exists within the muscle cell. And that process is indicated here. It is called glycogenolysis. Lysis, again referring to breakdown of something, and that something is glycogen. Glycogenolysis, so breakdown of glycogen. So one glycogen can be broken down to provide multiple glucose 6-phosphate. Okay, so for simplicity, we have one single glucose 6-phosphate here, and it can be either derived from glucose through this one reaction, or it can be derived from glycogenolysis, from glycogen. Either way, this is the starting point. So let's start with one glucose 6-phosphate. Now, for the sake of the exam, you do not need to memorize each of these reactions denoted by an arrow. You do not need to memorize all these terms other than a couple that I will point out and emphasize later on. Now, as you can see, I even left out the enzymes that catalyze each of these reactions. So you do not need to memorize every single reaction. So just focus on the ones that I emphasize. I just need you to generally understand this pathway and obviously the products of fast glycolysis. So from glucose 6-phosphate, which again is a starting point, we have another reaction that converts it to fructose 6-phosphate. From here, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. And as you see here, this reaction expends another ATP. So if the glucose 6-phosphate was derived from glucose, you can see that the net ATP yield is -2 at this point. If glucose 6-phosphate was derived from glycogen, you have a net ATP yield of -1 at this point. So -1 to -2 net ATP at this point right here. Now from here, you can see it branches off into almost 2 identical pathways. So everything is doubled from here on out. So as we get to this reaction here, which is again duplicated on this side as well, you can see that there is something going on here involving this molecule called NAD. And you can see that it is converting through some reaction to NADH plus H+. Now this NADH and this H+, which is just a proton, we'll talk about this later, is utilized in the aerobic system, which we will eventually get to. So let's hold off on the discussion on that. But let's discuss what is this reaction happening here. This reaction, again, on both sides here, represents something called a redox reaction, REDOX reaction. This is short for reduction oxidation reaction. This means one molecule is undergoing reduction while the other molecule is undergoing oxidation. So what is the difference between reduction and oxidation? Reduction is when a molecule gains electrons from another molecule, and the molecule that loses the electrons was oxidized. So again, reduction is when a molecule gains electrons from another molecule, and the molecule that lost those electrons was oxidized. So in this reaction here, NAD, which stands for nicotinamide adenine dinucleotide, is the molecule undergoing reduction because it gains electrons. It gains electrons from 3-phosphoglyceraldehyde, which is the molecule that is being oxidized since it lost the electrons to NAD. So in summary, NAD oxidizes 3-phosphoglyceraldehyde, and in turn, NAD is reduced. It gains those electrons. Now, what does it reduce to? As I said, NAD is reduced because it gains electrons, more specifically, 2 electrons. and it gains these electrons in the form of hydrogens. One hydrogen has one proton and one electron. So NAD gains two hydrogens. Now don't get too caught up in the details here, but the reason why it is NADH plus H plus is because the two electrons go onto this hydrogen here that is attached to NAD. And the proton that is left is dissociated, and that is denoted here as a single positively charged hydrogen ion, or simply a proton. So like I said earlier, the NADH with the two electrons and this proton is utilized for aerobic metabolism. So we will get to this again later. For now, let's move forward with glycolysis. Now, after this reaction, we see on both sides that the next reaction produces ATP. So at this step of glycolysis, two ATPs are produced. And remember, the ATP is produced because energy was released from the fuel molecule and used to form ATP, which will contain that energy in the phosphate bond. So this reaction here on both sides involves just that. So what is the net ATP yield at this point? Well, starting with glucose, you have 0 net ATP since 2 ATPs were expended up here. If the glucose 6-phosphate was derived from glycogen, however, then it would be 1 net ATP at this point. Minus 1 ATP here plus 2 ATP here equals 1 net ATP. So at this point, it's 0 to 1 net ATP yield. Now we move forward in glycolysis, and the next ATP producing step is right here. As you can see, we gain 2 more ATP from phosphoenol pyruvate. So at this point, we have a net ATP yield of 2 to 3 ATP, and we have 2 pyruvates. So during fast glycolysis, or the lactic system, which again is only partial carbohydrate oxidation, so partial glucose 6-phosphate oxidation, the pyruvates are converted to a molecule called lactate, and that is the end. So the products of fast glycolysis, aka the lactic system, is 2 to 3 ATP and 2 lactates. As a point of emphasis again, this is partial carbohydrate oxidation. So partial oxidation of this carbohydrate molecule called glucose 6-phosphate, which again can be derived from glucose or glycogen. So the very fact that this is just partial oxidation is alluding to the fact that there is more energy left in this molecule for more ATP production, and that is going to involve aerobic metabolism. In other words, full carbohydrate oxidation, so full glucose 6-phosphate oxidation, involves the continuation from glycolysis into the aerobic system. But for now, we are talking about partial oxidation, which is completely anaerobic, meaning fast glycolysis and the production of 2 to 3 ATP from one single glucose 6-phosphate does not require oxygen, so it is anaerobic. So partial glucose 6-phosphate or carbohydrate oxidation ramps up during post-anaerobic threshold exercise intensities. So 2 to 3 ATP very fast through these 10 reactions, ending in lactate. Through fast glycolysis, you are only getting a portion of the energy released from this glucose 6-phosphate, but you're getting it very fast. Okay, so here's the summary of net ATP yield through the lactic system or fast glycolysis. Starting with a single glucose 6-phosphate derived from glucose, you have -1 ATP from the start since it requires an ATP to convert glucose to glucose 6-phosphate. Then you have another ATP expending reaction, putting you at -2 ATP. Then you gain 4 ATP through the rest of glycolysis, and you get a net yield of 2 ATP. Now, starting with the glucose 6-phosphate derived from glycogen and glycogenolysis, you skip that reaction that glucose requires to be converted to glucose 6-phosphate. So here, that initial ATP expenditure does not occur. So then you only expend 1 ATP initially and gain 4, yielding a net of 3 ATP. And this is why we say through the lactic system or fast glycolysis, you get 2 to 3 ATP. And that is how it will be presented on the exam, 2 to 3 ATP through the lactic system or fast glycolysis. So if I ask what is the net ATP yield from 1 glucose 6-phosphate through the lactic system or fast glycolysis or partial oxidation, the answer is 2 to 3 ATP. So those three things, the lactic system, fast glycolysis, partial oxidation or partial carbohydrate oxidation, they all mean the same thing. So please make note of that because I can use any of those terms interchangeably during the exam. Now, one of the reasons why carbohydrates is considered a very versatile fuel source for muscle, especially during exercise, is that it can provide ATP anaerobically and aerobically. We just discussed how it provides ATP anaerobically through the lactic system or fast glycolysis. I also alluded to the fact that since fast glycolysis is partial oxidation, that there are other systems that oxidize the rest of glucose 6-phosphate to gain more ATP out of it. And that system is the aerobic system. So as represented here by the white area under the curve, a good portion of aerobic ATP production is through the remaining oxidation of the glucose 6-phosphate after glycolysis. And as you can see indicated here, the full oxidation of glucose 6-phosphate yields 32 to 33 ATP. This means that the aerobic system can produce 30 more ATP in addition to the 2 to 3 ATP that was produced by glycolysis. The oxidation in the aerobic system, of course, requires oxygen, while the initial glycolysis portion does not. So as intensity increases more and more, full oxidation of glucose 6-phosphate ramps up. But then as intensity exceeds the anaerobic threshold, we now have a shift in carbohydrate or simply glucose 6-phosphate oxidation. As you can see by the white curve, most of the glucose 6-phosphate that is available will undergo full oxidation through glycolysis, followed by the aerobic systems. But an increasing portion of the glucose 6-phosphate that is available will undergo only partial carbohydrate oxidation through fast glycolysis or the lactic system. So an increasing portion of glucose 6-phosphate will only yield 2 to 3 ATP and end in lactate, thereby not undergoing aerobic metabolism. So as far as carbohydrate oxidation is concerned, during post-anaerobic threshold exercise, the muscle cells increase the anaerobic portion of carbohydrate oxidation, which again is glycolysis. So to summarize, and this is what you should take careful note of, during post-anaerobic threshold exercise, there is increased full oxidation of glucose 6-phosphate, but an increasing portion of the glucose 6-phosphate is partially oxidized through the lactic system / fast glycolysis. And to answer why, well, it's because glycolysis does not require oxygen to produce ATP. Now in this slide, we can see this dynamic from a different visual perspective. So let's first turn our attention to the labeling on the left. As you can see here, full carbohydrate oxidation, so full oxidation of glucose 6-phosphate involves anaerobic ATP production, which is glycolysis, plus aerobic ATP production, which as you can see here, comes after glycolysis and occurs in the cell's power plant, the mitochondria. This is where aerobic ATP production occurs. Now, what else do you see in the aerobic portion? You can see that there is 30 additional ATP produced by the aerobic system. So with full oxidation, there is 2 to 3 ATP produced anaerobically without oxygen through glycolysis, and 30 more ATP produced aerobically in the mitochondria with oxygen. So when glucose 6-phosphate is fully oxidized, meaning all the energy is released from the glucose 6-phosphate, you get a total of 32 to 33 ATP. 2 to 3 ATP plus 30 ATP. That's 32 to 33 ATP yield. Now, what about fast glycolysis, aka the lactic system, aka rapid and partial carbohydrate oxidation. As you see by the labeling on the right, partial oxidation involves glucose 6-phosphate ending at glycolysis and not proceeding through aerobic metabolism in the mitochondria. You get 2 to 3 ATP quickly without the need of oxygen. So this portion again is anaerobic, and pyruvate does not proceed to aerobic metabolism in the mitochondria. Instead, pyruvate is converted to lactate, and that is the end for that glucose 6-phosphate. So essentially, lactate is simply a partially oxidized glucose 6-phosphate, meaning lactate has 30 ATPs worth of energy still left in it. And this is why lactate can be used as a fuel molecule. More on this in a second. So here's an important summary and takeaway for your note taking. During pre-anaerobic threshold, so exercise intensities below the anaerobic threshold, since there is sufficient oxygen availability to meet the oxygen demand, the aerobic system can fully oxidize glucose 6-phosphate after glycolysis. So meaning all the pyruvate that is produced from glycolysis does not convert to lactate, but rather enters further aerobic metabolism in the mitochondria for more ATP production. This means most of the glucose 6-phosphate undergoes full oxidation, which involves glycolysis, followed by aerobic metabolism, yielding 32 to 33 ATP per single glucose 6-phosphate. Again, this is characteristic of carbohydrate oxidation during pre-anaerobic threshold exercise intensities. Now, what about during post-anaerobic threshold exercise intensities? Since there is insufficient oxygen available to meet the rising oxygen demand, the muscle cell now relies on anaerobic systems to cover simply the ATP demand that the aerobic systems can fulfill. So the muscle cells ramp up only the anaerobic portion of carbohydrate oxidation, which is just glycolysis, while the aerobic system is reaching its capacity, meaning the mitochondria and the aerobic system cannot further process any more pyruvate. So there's an increasing portion of the pyruvate converting to lactate. So an increasing portion of the glucose 6-phosphate ends in lactate, providing only 2 to 3 ATP quickly per glucose 6-phosphate. In other words, during post-anaerobic threshold exercise, full oxidation of glucose 6-phosphate is still increasing, but because the aerobic system is reaching its capacity due to limited oxygen availability, the anaerobic portion, which is glycolysis, can ramp up ATP production further to help meet the ATP demand. This means an increasing portion of the glucose 6-phosphate is partially oxidized through the lactic system or fast glycolysis. Okay, so if I ask the test question, which of the following is characteristic of carbohydrate oxidation during post-anaerobic threshold exercise, you have all of these answers being correct. 1. Increased portion of glucose 6-phosphate undergoing partial oxidation. 2. Increased fast glycolysis or lactic system activity, which is the same thing as the first answer choice. 3rd, increased lactate production, which occurs because the fast glycolysis or lactic system is increased in activity. 4, increasing full carbohydrate oxidation with greater increases in partial carbohydrate oxidation, which is basically saying that this and this is both highly active during post-anaerobic threshold exercise. So all 4 of those answer choices would be correct. So as I emphasized, during post-anaerobic threshold exercise, there is increased lactate production because there is increased reliance on fast glycolysis to help meet the ATP demand. So yes, lactate does increase and build up during heavy exercise. Well, most of us have heard of the term lactate in some way or the other, And we generally know that lactate has a negative connotation associated with it, such as lactate causes soreness, lactate causes the burning pain during heavy lifting, lactate can build up and cause muscle pain, you need to flush out lactate for muscle recovery, and so forth and so forth. And I'll be straightforward with you and say all those sayings are not based on any kind of scientific fact. Let's discuss. First, as I said before, lactate is simply a partially oxidized glucose 6-phosphate molecule, which we know is derived from glucose as well as glycogen. So one way lactate is used by the body is for glucose recycling, meaning lactate can be used to be reconverted back to glucose 6-phosphate and then to glucose. And about 20% of the lactate formed during exercise is used in this manner. Now, the process by which lactate is recycled back to glucose is called the Cori cycle. And here's an animation to demonstrate this process. So here we have glucose circulating in the blood. Through muscle contractions or through insulin, the glucose can be taken up by the muscle cell and quickly converted to glucose 6-phosphate, as we know. Now, through glycolysis, we know that glucose 6-phosphate will provide 2 to 3 ATP, and this pyruvate will end up converting to lactate, especially during post-anaerobic threshold exercise. That lactate can then be shuttled out into circulation. From here, it can be taken up by the liver, and in the liver, the lactate can be reconverted back to pyruvate. And pyruvate can enter a process that is the reverse of glycolysis, which is gluconeogenesis, formation of new glucose. And through this process, pyruvate is reconverted back to glucose. And keep in mind, as I said earlier, this only occurs in the liver and not the muscle. Now, some of you may be wondering, why can't the lactate that is produced in the muscle cell be reconverted back to glucose in the muscle cell? Well, this is because the muscle cell does not have the enzyme to convert glucose 6-phosphate back to glucose. Only the liver does. This is why lactate must travel up to the liver to undergo conversion back to glucose. Again, this is called the Cori cycle. And the next slide is just a non-animated version of this slide. So to reiterate, about 20% of the lactate produced by muscle during exercise is used in the Cori cycle to be reconverted back to glucose. So what about the rest of the lactate? Well, like I said, lactate is a partially oxidized glucose 6-phosphate, or carbohydrate to be general. Remember, only 2 to 3 ATPs worth of energy was released from the glucose 6-phosphate before lactate was produced. Also remember, if pyruvate was not converted to lactate and continued on into the aerobic system in the mitochondria, 30 more ATP would be produced. This means one single lactate has about 30 ATPs worth of energy still left. I'll repeat that, lactate has about 30 ATPs worth of energy still remaining because, again, it is a result of fast glycolysis, which again is only partial oxidation. So this means lactate is a very useful fuel molecule. So lactate can be picked up by muscle cells and reconverted back to pyruvate. From there, pyruvate can then enter the aerobic system after converting to a molecule called acetyl-CoA in the mitochondria. More on that later. And you get 30 more ATP from that lactate. This is usually what happens during the time after high to max intensity exercise. So say during high to max intensity exercise, your muscles have high activity of the lactic system and there is increased lactate production. Say you follow that high intensity bout with some low intensity exercise like a cool down or a light interval during HIIT training. During that light interval or cool down, you are exercising at a pre-anaerobic threshold intensity. So now the lactate can be used to produce ATP aerobically. So main takeaway is that a majority of the lactate formed during exercise is used as a fuel molecule because it still has 30 ATPs worth of energy left. The body does not like to waste valuable fuel molecules. That's like any biological organism. If there is a fuel molecule present and it is a source of energy, the body cell will utilize it. So lactate is not just simply a waste product as many people describe it to be. It is commonly referred to as a metabolic waste product. As you can tell, it is not. It is a valuable fuel molecule that your cells can utilize, especially muscle cells. So just to reiterate, the lactate that was formed can eventually be reconverted back to pyruvate, and then that pyruvate can enter the aerobic system for further oxidation and ATP production. So if we go back a couple slides here, we are talking about lactate reconverting back to pyruvate, and then pyruvate moving forward into the mitochondria for further aerobic ATP production. And this is why we said that lactate has about 30 ATPs worth of energy left. So most of the lactate that is produced, especially during post-anaerobic threshold exercise, will eventually follow this pathway right here, from lactate to pyruvate to aerobic ATP production. It is a valuable fuel source. So what this means is that lactate does not just build up or get trapped inside muscle cells. It does not just hang around and causes problems. It is not a villain, in other words. It doesn't cause fatigue. It doesn't cause the burning sensation. It doesn't do anything other than provide energy for cells in the body, such as muscle cells, or provide substrate for glucose production. So again, it is a source for glucose recycling through the Cori cycle, or as a fuel source through aerobic metabolism. As I alluded to earlier, one of the major myths associated with lactate is that it is the cause of muscular fatigue and pain during exercise, often described as the burning sensation. This is false. As shown in this figure, the lactate produced during heavy exercise or post-anaerobic threshold exercise, as discussed earlier, is used as a fuel for ATP production or substrate for glucose production via the Cori cycle. So what is the cause of muscular fatigue and pain during heavy exercise, like during a set of heavy squats? It's not lactate or lactic acid, which, by the way, we do not produce lactic acid during exercise. We produce lactate. They are not the same thing, even though many will tell you it is. The cause of fatigue during heavy exercise and also the pain is mainly acute metabolic acidosis. So what is acute metabolic acidosis? Well, when we exercise post-anaerobic threshold and as we get closer to max intensity exercise, there is a very high ramping up of two particular things. We have a very high rate of ATP hydrolysis since muscles are contracting at a high level. And we also have a very high rate of redox reactions, which we discussed earlier with glycolysis. Both of these processes produce positively charged hydrogen ions, which again is simply called a proton. Usually these hydrogen ions are cleared out by the mitochondria because they are used in the mitochondria for the aerobic metabolism. But during post-anaerobic threshold exercise, remember the aerobic system and thus the mitochondria is reaching its capacity and it is almost quote-unquote tapped out, so to speak. So it can't take in and process any more of these protons, among other things. So the protons will start to accumulate in the muscle cell's sarcoplasm, which is just basically the intracellular space. And when protons start to accumulate in solution like the sarcoplasm, the solution would become more acidic, as denoted by a decrease in pH. So overall, muscle cells become acidified and muscle pH decreases. Now with any cell in the body, when pH is disturbed, like when it becomes more acidic, the cell starts to become more dysfunctional. Why? Well, because cellular processes can only work within a normal pH environment. So when muscle cells become acidic, processes like cross-bridge cycling and muscle contractions becomes inhibited. They don't work very well in an acidic environment. So if contractions are being inhibited due to this acidic environment, then how does that affect performance during, say, the heavy squats? Performance will start to decline. As you perform rep after rep, what happens? You feel weaker and weaker. You squat slower and slower no matter how hard you are trying. Again, this is because the acidosis is inhibiting muscle contractions. Now sometimes, say like during the heavy squats, one of the factors that contribute to fatigue or this decline in performance is pain or burning sensation. This pain is caused also by acute metabolic acidosis and the buildup of protons in the muscle cells. Sensory neurons that carry pain signals to the brain are triggered by proton accumulation. So this is why the muscle burns, quote unquote, when you are performing heavy exercise. Now another factor contributing to fatigue that is not through acidosis is the inorganic phosphates. Remember, one of the products of ATP hydrolysis is the last phosphate that broke off of ATP. This phosphate is usually processed in the mitochondria to be used to rephosphorylate ADP back to ATP via aerobic metabolism. But again, the mitochondria and the aerobic system has reached its capacity. It is again, quote unquote, tapped out. So the inorganic phosphates from the rapid hydrolysis start to accumulate within the muscle cell. This has also shown to impair muscle contractions. So in summary, fatigue during heavy exercise is not caused by lactate buildup. It is caused by acute metabolic acidosis from proton accumulation and also the accumulation of inorganic phosphates within the intracellular space. Now in my KIN4540 Exercise Bioenergetics and Metabolism course, we will discuss why we have this thing called fatigue and why we have pain during post-anaerobic threshold exercise, especially max intensity exercise. What is the purpose of fatigue and pain during, say, a heavy squat set? So these questions will be answered and we will understand the biological reason for this thing called fatigue. So more on this in KIN4540, so stay tuned for that course.